Let's see. All right, so chapter 14 is our chapter focusing on genetics, and that's pretty much what 14 and 15 are. They focus heavily on genetics. But you have to understand the process of type of cell division involving meiosis that we covered in chapter 13 to help you understand what's covered in chapters 14 and 15. Now, chapter 14, titles Mendel and the gene idea, but what it is Mendelian genetics. Mendelian genetics focuses on patterns of inheritance, how traits are inherited from one generation to the next. Now, let me get the little hand here. So this chapter, you can relate it to everything from evolutionary adaptation to reproduction to growth and development. Now, I have a few slides that are just review. I'm going to try to go through these a little quicker than what I did this morning, but we talked about how in the type of cell division involving meiosis, how the gametes that are produced, the sperm and eggs that are produced, okay, usually are not identical. As a matter of fact, you can think of it as 99% of the time, 99.9% of the time, they're not going to be identical. And that is due to what happens in meiosis 1 to begin with. In prophase 1, we have crossing over, which results in DNA recombination. This is where homologous pair of sister chromatids can exchange DNA and eventually results in a sperm or an egg carrying, if you want to call it, a hybrid chromosome. It's a mixture of maternal and paternal DNA, which you don't see in any other cell in the parent's body, but can be passed on to the child. Now you'll find it in every cell that makes up the child's body. But then we talked about metaphase 1 and meiosis 1. And this had to do with independent assortment. How during meiosis 1, specifically metaphase 1 of meiosis 1, the homologous pairs of sister chromatids pair together forming tetrads, and they will either align to where just by chance all the paternal DNA is attached to the spindle on one side and all the maternal DNA is attached to the spindle on the other side. However, how this tetrad here made up of a homologous pair and how this tetrad right below made up of a homologous pair, how they align in terms of which side the maternal and the paternal DNA is positioned, it's independent as they're showing you here. They could align this way or they could align this way to where we have both maternal and paternal, okay, facing in both directions. We call that independent assortment. Okay, in this case, what that means is, yeah, there's a possibility that the one set of 23 you inherited from mom, you happen just by chance to pass those on to your child. However, it's more than likely that the 23 or that one set of chromosomes you pass on to a child or to an offspring is going to be a mixture. It's going to consist of both maternal and paternal chromosomes that you inherited from your parents, and that's on top of the crossover event, which could also lead to a hybrid chromosome being passed on. What contributes to genetic variation even further is that fertilization is random. Okay, many sperm are being produced. Only one's going to be involved in the fertilization process of an egg. Now, as far as eggs go, granted, it's a little different for women, but the bottom line is whichever egg is released during ovulation, well, it's at random. It depends which one, so therefore the genetic information carried in each set of 23 chromosomes that each gamete has, that's going to vary. And that's going to lead to a greater amount of genetic variation in the offspring. But the greater the amount of genetic variation within a population, a group of the same species, the greater the chance that population can evolve and adapt. So it's usually considered a good thing. However, that could lead, okay, to two possible combinations, especially if you have brothers and sisters. It could lead to where if you have brothers and sisters, okay, some of y'all look more like one parent than the other parent. It's possible that you may look a little bit like both parents or neither parent. Okay, I used to tell my brother he was adopted because he said he didn't look like either parent, but the truth of the matter was that no, he wasn't the milkman's child. Excuse the pun, that's an old saying. But you can see here, if you're familiar with a comedian named Damon Wayans, you're probably more familiar if your younger generations with his son, Damon Wayans Jr., such as in the movie Cop Out. They look a lot alike. The son looks a lot like the dad. Over here, Reese Witherspoon. This is her here, and even I forget. That's her daughter over here. They look a lot alike. So you can see the physical traits that are passed on. Now, granted, the parents may have a lot of genetic variation between themselves, just looking at their phenotype or their physical features. Okay, and that can lead to a greater amount of variation in their children. However, chances are you can still find, say, at least physical traits that match up with one or both parent. Okay, So this is what we're going to be focusing on this chapter. It's genetics, but it's patterns of inheritance. The term genetics itself, roughly coined around the turn of the century in the 1900s. As we transition to the 1900s, they say approximately around 1905 to 1910 is when the term was actually coined. But genetics is defined as the science of heredity. Now, I don't know if you've ever done this before, but you don't know the meaning of a word, so you look up the definition of the word, and in the definition, they give you another word that you have to go look up. It's like, great, this is just leading to more problems. Well, that's the way it works for genetics. Somebody gives you the definition of genetics, science of heredity, then for most people, they're like, okay, then how do I define heredity? And that's down here. Heredity is a process by which specific traits are passed on from parent to offspring through genes. Okay, now we're going to go over the experiments conducted by the man who's considered the father of genetics, Gregor Mendel. Except for Gregor Mendel, he was only going by observable traits, which are also known as phenotypes. At the time he conducted his experiments was in the 1850s to the 1860s and published his work around 1866. However, the compound DNA had roughly around that time in the 1860s been discovered, but it wasn't called DNA. All scientists knew is they had something that looked unique. By the 1870s, a chemist had uncovered some of the structures such as the bases associated with DNA. However, by the early 1900s, when they established, okay, this is a new compound we have, DNA, the majority of the scientific community did not believe it contained what we call genetic information. But the term genetics, the term genes, genotypes, all that had been coined, but they didn't know what compound they could directly apply those terms to. Many scientists thought it had to be proteins that were responsible for the passing of traits, okay, from one generation to another, such as in the fruit fly, better known as Drosophila melanogaster in the scientific community. Okay, they're known to have red eyes or white eyes, and we'll go over this more in chapter 15 about them. Okay, and it's found, hey, the color eyes that the organism has, well, it's passed down from one generation to the next generation to the next generation. Okay, that was the phenotype, or the trait is eye color, and there's two different phenotypes, red and white. It wasn't until roughly just past the 1950s, roughly around the mid-1950s to the 1960s, that the scientific community got on board, everybody, and said, okay, we thought, those that thought it was proteins, it's because there were so many different proteins out there. They did so many different types of varieties of works, it had to be them. By the early 1900s, as they uncovered more and more about the structure of DNA, one thing they did come to find out for DNA is, hey, there are 4 bases that contribute to the formation of DNA, but that's too simple. There's no way that this compound DNA could lead to so much diversity, but you have to think of it this way: less is more. Okay, and that's what they eventually find out. And we'll visit this later again after the 3rd exam about what's one of the experiments that finally led to conclusively proving genetic information is stored in DNA. It's not proteins. Okay, so it's just showing you here, that's what this figure is showing you. Now, I show you this here, sister chromatids, and if you recall, chromatin, chromosomes, sister chromatids, and now genes or alleles. When you hear any of those terms, the compound we're referring to is DNA. Okay, but what scientists found is as they scan the DNA, and of course with additional information, as they scan the DNA, eventually they realized there were segments or regions of DNA that formed units of genetic material called genes. Okay, so this slide that I'm on right here, I believe it's number 9, is there to feed directly into the next slide, number 10. The way it works when we talk about genetics is we cheat a little bit. Okay, it's easier to visualize DNA tightly compacted as a chromosome, even though technically if you go on to advanced classes or grad school, you'll find out DNA only tightly packs together and sit in the form of a sister chromatid or chromosomes when a cell is dividing. However, we cheat, just like we cheated a little bit with the karyotype, and we say it's a chromosome map. We cheat a little bit here to simplify the explanation about genes and genes that are called alleles. But first you have to know chromosomes are DNA molecules. They're showing you a chromosome right here. Now the chromosome, you can see here, they've given it different banding, a different banding pattern. Some of the bands are thick, very thick. Some are very thin. Some are dark colored. Some are light colored, especially right here where the centromere is. Well, what it is for geneticists is that they would scan DNA that conform to different chromosomes in our cells. And as they scanned them looking for genetic information, eventually they would come across regions, such as, say, I'm just arbitrarily circling this. They would come across regions, okay, that would contain information, genetic information. And they would coin that region or unit or segment of DNA a gene. Now, where that gene is located on the chromosome, we call that the gene loci. As far as the gene itself goes, this is not 100% true, but for this class, it's good enough. Okay. For now, I want you to think of genes as protein recipes. Okay. Now, a geneticist or a genetics teacher, if you take a genetics class, they'll say genes code for proteins. There is another compound they'll code for, but like I said, we're going to just keep it this way, simplify it. So genes code for proteins, but that's another way of saying protein recipe. What it is we talked about proteins do the work in a cell. So what happens is when a cell needs a particular type of work to be done and the protein's not present, the cell will go and find the gene loci for the gene it needs on the chromosome it needs, okay, or find the chromosome that has a gene loci containing the gene that contains the information on how to make the protein to do the work the cell needs. And we're going to talk about this later in chapter 17. We're going to talk about gene expression. Okay. Sometimes when a cell needs a certain type of work to be done, the protein it needs to do the work is not present. So what the cell will do is go back and look at its genetic information. It'll find the chromosome that has the gene loci containing the gene that it needs, so then that it can cook up the protein or express that recipe and make the protein. So now it has what it needs to do the work. So then, as if that wasn't complicated enough, they started referring to genes. As they became more and more familiar with genes, they started to find out, well, when you do a karyotype, initially when you see the karyotype, if you recall, the chromosomes, we have 46 chromosomes, but they're paired together. And in humans, whether it's female or male, okay, at least 22 at a minimum, at least 22 out of the 23 pairs are definitely homologous chromosomes known as autosomes. Okay. Now, when they're paired together on a karyotype, they're paired up with chromosome pair being the longest, the largest, all the way down to chromosome pair 22. Those homologous chromosomes are very small. Initially, when you look at that karyotype, you know, the length of the chromosome should stand out and where the centromere is located. So structurally, they're homologous. They're similar or the same. However, for a geneticist, they say, no, there's more to the story. They're not just homologous because they look the same. Their appearances are the same. What really makes them homologous is the genes they carry. What geneticists found is as they scan one of the two homologs, when they come across a region or segment of DNA that forms a gene located on that chromosome, which we call the gene loci, on its homologous pair, they don't have to scan the DNA to look for a similar gene. All they have to do is go across to the homolog at the same location, at the same gene loci, and they found that they will find a similar recipe. Similar genetic information will be located at the same gene loci on the homologous pair of chromosomes. And this genetic information will code for the same trait, such as pea plants, flower color. That is one trait, flower color. However, there are two different colors or two different phenotypes for flower colors we'll talk about. It could be a purple flower color or a white flower color. So down here, you can see the reason why they call genes alleles. Well, for geneticists, that infers by calling a gene an allele, that infers to others familiar with this discipline, that what they're saying is alleles are one of two or more alternative forms of a gene that may arise by mutation. They'll be found at the same gene loci as I showed you here on a homologous pair of chromosomes, except those two or more alternative forms of the gene that we call alleles, they code for the same one trait. And as we go through Mendel's experiments, I will talk about how there are 7 traits he studied. You're going to be responsible for 3. And for those 3 traits you're responsible for, they come in 2 different phenotypes, a dominant and a recessive phenotype. Okay, but what phenotype the organism or what observable feature you see in the organism, it's determined by what we call the genotype, which I'm circling here. So through Mendel's work, what geneticists eventually found is not only for many traits, okay, of an organism, many observable features of an organism, not only are they encoded for by genes that we call alleles, but there are often two or more forms of that gene or alleles that code for that same one trait. And what they found is sometimes the genetic information in one allele, okay, tends to dominate genetic information in the other allele by determining the phenotype. Okay, so eventually what they started doing is they started referring to these alleles, okay, as dominant and recessive alleles. Okay, whenever we have at least two or more different forms of a gene that code for that same one trait, we not only call them alleles, but if one of those two forms of the allele, okay, if all it takes is to inherit just one of those two, that how do you say, one copy of that allele to determine your phenotype, then we refer to that allele as a dominant allele. Okay. Now, sometimes what happens is that an offspring will inherit a homologous pair of chromosomes, and at the same exact gene loci on both chromosomes, both the alleles, okay, not only code for the same trait, but they may happen to both be what we call dominant alleles. They're dominant alleles because they're going to determine the phenotype, and therefore at the genotypic level, we refer to the person as being homozygous dominant. Now, if you know the names of the alleles, you just use two uppercase letters, and I'll explain where those come from in a bit. However, the way they determine if the two or more forms of an allele that code for the same trait, if the way they determine if one of them dominates the other, is by looking at individuals that we refer to as having a genotype heterozygous dominant. That means that when they inherited a homologous pair of chromosomes, one from each parent, at the same gene loci, from one parent, they may have inherited an allele that contains genetic information that will determine the phenotype. Whereas on the other homologous chromosome, they will have inherited an allele that codes for the same trait. However, it gets masked or hidden by the other allele. You can't see it in the phenotype. It's hidden. We call that the recessive allele. And the only time the phenotype is impacted by a recessive allele is if you inherit a homologous pair of chromosomes, okay, at the same gene loci. The hand's moving a little slow, so I'm trying to slow down. And the same gene loci on this homologous pair of chromosomes, you have a recessive allele on both. Therefore, there's no dominant allele. Okay, I'm getting connection problems here. Bear with me. All kinds of stuff is starting to flash across my screen now. Okay. Now, what happens is the only time you see the phenotype determined by genetic information stored in the recessive allele is if on both homologous chromosomes you inherit a recessive allele at the same gene loci, therefore for the same trait, and therefore you are considered, as far as your genotype goes, homozygous recessive. Okay. Now notice something. These chromosomes are in different colors, and I talked about this in chapter 13. You have to remember, not only for a cell division involving meiosis, but also when you get into Mendelian or genetics in general, you have to take into consideration the color, such as, let's say, red is maternal and blue is paternal. Okay, but we talked about this, right? During a life cycle with cell division involving meiosis, sperm and or eggs are being produced. These are haploid gametes. Each carries one set of chromosomes, and they should match up to where we end up with 23 pairs. At a minimum, 22 should be homologous. So again, when we talk about a pair of chromosomes being homologous, it's not just the structure. It's the genetic information they are carrying. They are carrying genes better known as alleles, because often you have genes for the same trait that are not 100% identical. So, and often one of the alleles is dominant and the other one is recessive. And it's just a matter of whether you inherit a pair of dominant on a pair of homologous chromosomes, a pair of recessive for the same trait on homologous chromosomes, or whether you're what we call heterozygous dominant. From one parent you inherited a dominant allele, from the other a recessive. Okay. So we talk about genotype and phenotype. I edited the PowerPoint so you can follow me hopefully a little better along with your notes, okay? When you hear the term genotype, automatically tell yourself you're talking about the genetic makeup of an organism. Okay, the genotype is every gene inherited by the offspring, or sometimes we'll say the genotype when we're only focusing on the genetic information, okay, that determines one trait, such as down here. We're going to go over Mendelian genetics. We're going to talk about monohybrid crosses. And in Mendel's, he didn't know any genes, alleles, no, those terms weren't even coined. What he did notice is that from one generation to the next, certain traits could be passed on, such as purple flower color from one generation to the next. What he also noticed is that sometimes a trait isn't passed on from one generation to the next. But as I'll show you, if he allows this next generation to produce another generation, sometimes what looked lost, okay, sometimes that one phenotype that looked like it was gone, it reappears in a generation further down the line. Okay, so, and then we'll talk about how geneticists came along. Once it was conclusively proven that DNA is a compound by which genetic information is stored, geneticists came along and identified the alleles, okay, that determine one trait for flower color. Except what they uncovered, okay, is that there are two forms of the gene responsible for that one trait flower color. One's a dominant allele and one's a recessive allele. Now this is the catch. We're going to talk about this more. For Mendelian genetics and focusing on his pea plant studies, okay, the dominant allele is given a name based on the dominant phenotype. So when we go over flower color, we're going to say how geneticists, when they identified the allele responsible for purple flower color, they simply took the P from purple, placed it as an uppercase in front of allele and called it the P allele, the dominant P allele. Okay. So again, sometimes the name of the allele comes from the dominant phenotype. But when they discovered the allele responsible for the other phenotype, white flower color for this one trait flower color, okay, they give that allele a name, still using the first letter from the dominant phenotype, but they use a lowercase letter, lowercase p, to denote that this allele that codes for white flower color, it's a recessive allele. Therefore, if you look down here, if there are any pea plant offsprings that are heterozygous dominant for flower color, just inheriting that one dominant P allele is enough for those offspring to produce purple flowers. The other allele, denoted by a lowercase, even though this allele codes for white flower color, we choose a letter based on the dominant phenotype, P for purple. This lower P, which represents the recessive allele, it's masked or hidden in this organism that's heterozygote. Now the phenotype, the way we're going to approach the phenotype, it's the observable features that make up the phenotype. Okay, that's heavily influenced, but not always solely determined by the genotype, as we'll talk about at the end of this chapter. I told my morning class, this is probably the one chapter, along with one or two others, that I would much rather do in person, so bear with me. On that note, before we go any further, I tried to post several video links. Some of the video links should not be more than 2 or 3 minutes, such as there's a video link for Punnett square, then there's a video link for monohybrid cross and for dihybrid cross, which are two types of experiments performed by Gregor Mendel. Okay, so watch those video links. They're the shortest ones I could find that focus directly on what we're doing here. There are some initial video links I believe I have on page 1. Okay, they're lengthier. They're about roughly 10 minutes, but just hang in there and read through those, please. Okay, the only time I get a call is when I'm doing this. Okay, so if this chapter is not pleasant, this is the guy to blame, is what I tell students. Okay, Gregor Mendel. PBS. Yes, actually, Ashlyn, a lot of the videos I gave you were Amoeba Sisters. Okay, but I found some that were shorter. Okay, but yeah, we had a department meeting. Some of the other professors were using Amoeba Sisters, but you know how it works. You can't always satisfy everybody. So some of these videos that are 10 to 12 minutes are actually, they fluctuate. For Amoeba Sisters, I've had them as short as 6 minutes, as long as 12 or 13. So But watch them, please. Watch them along with what we're going over here. Okay, so this is a man you have to thank. It's a monk by the name of Gregor Mendel. PBS had a documentary last week having to do with genetics, and they actually showed pics of Gregor Mendel at the monastery, and they were actually showing you all the pea plants he was growing. We're going to simplify, but I want to let you know right now, okay, the, how do you say, the data I present, the experiments I present, he actually performed these experiments hundreds, if not thousands of times. And I'm going to be giving you ratios, phenotypic ratio of 3 to 1, or a phenotypic ratio of 9 to 3 to 1. Okay, but in actuality, he was producing thousands, if not tens of thousands, of offsprings through breeding studies that you're going to hear me refer to as cross-fertilization or self-fertilization. And basically, he generated so many offsprings when he ran each experiment that it was going to be really hard for him to come up with a false negative or false positive. He wanted to ensure that he did a thorough job. Okay, so we go over Mendelian genetics and pea plants, but if you really take a step back and think about it, he was ahead of his time. And even for me, I wondered, did you just get that lucky guessing to use pea plants, knowing you had to produce this many offspring? Or are you just insightful? Are you just intuitive? Basically, are you a genius that just sees things other people don't do? Because he did a very thorough job. Very. Therefore, he is considered the father of genetics. As we'll talk about in 15 with another man who won the Nobel Peace Prize, Thomas Hutton Morgan. He also performed genetic studies focusing on the patterns of inheritance, but he did it with Drosophila melanogaster, the fruit fly. Some of his results using Mendel's experiments came out just like Mendel's for the pea plant. But some of his results contradicted what Mendel got. But he was feeding off of Mendel's work. And he was finding there are variations, which we're going to start visiting at the end of the chapter. So he is a father of genetics. He published his findings in 1866, and he provided evidence of discrete heritable units that would eventually be called genes. So this discrete heritable units and the term genes, these actually came about, okay, well before DNA was finally and conclusively proven to be the compound that contained genetic information. So the field is the discipline of genetics, terms such as, like I said, of genes, genotypes, alleles, all these terms are coined. They came up with these terms by the turn of the 1900s, the beginning of the 1900s. They just didn't know what compound to actually attach those to until later. So listen carefully. Mendel studied 7 traits. You are responsible for just these 3, which I'll also give you in your notes. So you can see here the traits he actually focused on were flower color, flower position, seed color, seed shape, pod color, and stem length. I'm only going to test you on these 3, and I put these arrows over here to try to reemphasize it. So listen, the way it goes, because you'll hear the term traits and then you'll hear the term phenotypes. Okay, and I would have students saying, you're saying traits are observable features, but I've always been told phenotypes are observable features. Okay, well, phenotype fits under traits. Okay, so we're going to focus mainly on flower color, and then I'm going to talk to you how you just pull and plug the information. Pull out the information that I go over for flower color, plug in the information for seed color. Pull that information out, plug in the information for seed shape when we go over one of his 2 types of crosses. But the way it works is this way. One of the 7 traits that he studied, the pattern of inheritance from one generation to another generation of pea plants, was flower color. Except pea plants can produce 2 different colored flowers. Therefore, there's 1 trait for flower color, but there are 2 phenotypes for this 1 trait. There's a phenotype that is now referred to as a dominant phenotype, which is the color purple. But there's a second phenotype for this 1 trait, flower color. Okay, it's now known as a recessive phenotype for white flower color. So we have 1 trait for flower color, but it comes in 2 different phenotypes, a dominant purple or a recessive white. If you come down here, seed color, this is another trait. But Mendel found that seed color also comes in 2 different phenotypes. There's a dominant yellow colored seed and a recessive green colored seed. So these are the 2 different phenotypes. And then there's a third trait, seed shape. This is a third single trait, seed shape, but it also comes in 2 different phenotypes. Okay, a dominant round shape and a recessive wrinkled shape. So this is the best way I can do it, especially, usually I do it on the board, but a dry erase board, but I can't write worth a darn on this thing, this white erase board here. So hopefully the way I've labeled this makes a little sense, okay? So we're going to focus on these three traits, but I'm heavily going to focus on flower color for one type of experiment Mendel performed, and then I'm going to use these other two traits for a second type of experiment that Mendel conducted. Okay. Now, I haven't come across, but I haven't done extensive background work searching everywhere, but Mendel choosing plants in general, and not just any plants, but what we call flowering plants. When we go into 1407, if you ever take 1407, at least in my class, we go over plants and how 350,000 of the roughly 390,000 species of plants that exist today, they're flowering plants. Okay, and then just to give my students their money's worth, we talk about how a flower is actually a reproductive structure. Okay, a flower consists of a carpel, which is where eggs are produced, and that's shown down here. This little bowling pin looking thing, that is the carpel that functions as a female part. That's where eggs are produced. But a flower also consists of what we call a stamen, and this is where sperm are produced. And you can see here they're showing you multiple stamen here with these long little thin filament looking thing, which is actually called a filament, going to this expanded tip, which we call an anther. Okay, and it's at the anther, or these little yellow tips they're showing you here, where sperm are produced. Now, sperm are actually encased within pollen. So when you hear about pollen being dispersed, it's what's in the pollen contains sperm that allows it, if it lands on the same flower right here at the carpel or a different plant, allows fertilization to occur. Now, what I mean by Mendel choosing plants, what was a great thing about plants is plants can reproduce two different ways. Okay, they can asexually reproduce, whereby it only requires one parent who has the ability to produce both egg and sperm. Except I'm not going to say asexual reproduction when I go over Mendel's work. I'm going to say self-fertilization. Because a flowering plant produces flowers that have both a carpel and a stamen, that means that one plant can self-fertilize itself and asexually reproduce. One parent produces offspring. However, Mendel also knew that plants can sexually reproduce and that he could control both processes. Okay, he just had to be careful, but he could control them. Well, when plants sexually reproduce, you're going to hear me say cross-fertilization. Mendel would start his pea plants, okay, as we'll talk about in a bit, with pea plants that were called true breeds. And what he would do would manually transfer some of the pollen containing the sperm from the stamen of one pea plant over to the carpel, which contained the eggs of another pea plant. So now there's not one parent like in asexual reproduction or self-fertilization. There are now two separate parents, and they're undergoing sexual reproduction, or what I'm going to call cross-fertilization. The advantage to Mendel using plants, okay, was it allowed him to limit, okay, what traits could possibly be passed on from one generation to another, such as what we'll talk about. Over here it says P. We're going to talk about how when Mendel performed his crosses, okay, the initial plants he used made up what we call the P generation, the parent generation, and the offspring they produced were known as the F1 generation. Okay, if he cross-fertilizes, sexually reproduces, he's increasing the amount of genetic variation that can be passed on to the F1 generation, which we call hybrids. However, we'll talk about how from the parents to the offspring, one of the two traits that the parents has is gone. Mendel takes advantage of the fact pea plants can self-fertilize to investigate if each individual pea plant might be carrying hidden information for the trait that disappeared, which is going to be white flower color, as we'll talk about. Okay. So Mendel's experiments consisted of two types. The first type was called monohybrid crosses, and the second type, they're called dihybrid crosses. So listen carefully. The cross for both types of experiments has to do with the first type of fertilization he's going to perform, which is cross-fertilization. In other words, it's going to require two separate parents. Okay. The mono, M-O-N-O, that prefix, okay, I define as focuses on one trait. Okay, so the mono, M-O-N-O, you can define it as focuses on one trait. And what do I mean by focuses on one trait? Focuses on the pattern of how one trait is passed from one generation to the next generation to the next generation. That's the mono. The hybrid, everybody, the hybrid is in relation to the offspring produced by the P generation, which is the F1 generation. So the hybrid is in reference to the F1 generation, because as we'll talk about, each F1 generation offspring, it's a combination of two separate parents. It's a hybrid of both parents. Okay? I'll talk about dihybrid when we get there. So for monohybrid crosses, what I'm going to focus on is flower color, but at the end I'll briefly point out how you can just pull and plug information and replace the flower color, the trait for flower color information, with seed color or seed shape. Okay, now as if that's not tough enough already, what Mendel actually did before he ever started any of his crosses was he first generated what are called true breeds. True breed is a way of saying pure breed. Okay, except for plants, he called them true breeds. And the way he generated these true breeds was through self-fertilization. Okay, so ignore all this around here and just look at what I'm circling. What he did is he would take a pea plant that he saw produce purple flowers, and he would allow it to self-fertilize and produce offsprings. Then he would take those offsprings, all those offsprings, and allow them to self-fertilize and produce their offspring, until eventually he noticed that for some of these plants that were self-fertilizing, they were always producing plants that grew the same colored flowers, purple. He would isolate those plants, and that proved he had a true breed. So once he had a true breed, if it was a true breed for purple flower color, every time he let that plant self-fertilize itself, it always produced offspring with purple flowers. Same thing for this other one here. Before he ever started the monohybrid cross, he also created a true breed for white flower color. That meant if this true breed here for white flower color, if it self-fertilized, the offspring that were produced always grew white flowers, always. So once he established these true breeds, then he began his monohybrid cross. He took these two separate true breeds that differed in the trait flower color. One was a true breed for purple flowers, the other was a true breed for white. These two plants made up his P generation, the parent generation. And then what he did is he took the pollen from one plant and used it to fertilize the eggs in the carpel of the other plant through a process called cross-fertilization. He did this several times over. So he performed many monohybrid crosses just for the trait flower color. And no matter how many times he did it, the offspring produced by the P generation, we call these offspring the F1 generation. Okay, the F1 generation, they are the offspring of the P generation, and they are hybrids. Okay, they've inherited information or traits from two different parents that's combined into one. And no matter how many times he did this monohybrid cross for flower color, for the trait flower color, the F1 generation offspring always grew purple colored flowers. Yes. Yeah. It took him a long time. A long time. Yes, it did. I'll see if I can find that documentary, that PBS video just from last week. I don't know how much of an attention grabber it is, but they talk about his work towards the beginning. Okay, but yeah, I had another textbook I used to teach out of, and for genetics, I taught it for a semester, and oh God, they actually gave me the numbers, and it was in the hundreds of thousands overall. Okay, so what happened then is for that F1 generation plants, no matter how many times he took two separate true breeds and took them and cross-fertilized this P generation, producing the F1 generation offspring, all the offspring always developed purple flowers. So that led Mendel to question, what happened to the trait for white flower color from the parent from one of the two parents he used for this cross-fertilization. Because remember, this true breed for white flower color, if it self-fertilizes, it passes on the trait for white flower. But when you take it and cross-fertilize it with another true breed, but for the color purple, all of a sudden the trait disappears. So this is where it came in to play, that he could take advantage of plants having the ability to asexually reproduce or self-fertilize. What he did then next to find out if this trait for white flower color was gone, or if it was just hidden in each individual F1 plant, is he took this F1 generation, he separated each plant from the other, and he allowed each one to individually self-fertilize. And when that happened, he produced thousands upon thousands, if not tens of thousands of plants. But the way I describe it, when that happened, first of all, each individual F1 generation plant that self-fertilized produced offspring referred to as the F2 generation. And once Mendel had taken enough of these F1 generation offspring, allowed them each to independently self-fertilize and produce collectively a population of offspring known as the F2 generation, he then went about, recording the results as far as what numbers on average were producing purple versus which ones might be producing white. Well, he noticed that although it didn't happen frequently, he definitely had the resurfacing, if you want to call it, or the reappearance of that flower color trait, white, like he had in the initial P generation. So it skipped a generation. Then what he did, or what they say, is even though he produced tens of thousands of F2 generation offspring, on average, for every 4 plants he counted, 3 produced purple flower colors. And on average, for every 4 plants he analyzed or counted, 1 out of 4 would produce white flower color. Now listen, it didn't matter if he was studying the trait for flower color, seed shape, seed color. Okay? The F2 generation always resulted in a phenotypic ratio of 3 to 1. So again, it didn't matter which of the 3 traits we were to look at here. The phenotypic ratio for the F2 generation was always 3 to 1. And what do we mean by 3 to 1? On average, 3 out of every 4 produced the dominant trait for purple, or the dominant phenotype for purple, and 1 out of every 4 produced the recessive phenotype for white. So that was a ratio of 3 to 1. Well, like I said, it wasn't until about the mid-1900s, and actually pushing into the 1960s, that it was squashed. Scientific community was on board. There have been experiments conducted, one that we'll cover, that conclusively proved genetic information for living organisms is stored in a compound called DNA. And as technology improved and they learned how to isolate chromosomes from cells, what they did is for plant cells, they managed to eventually isolate a pair of homologous chromosomes, and they located the gene loci for the trait flower color. Again, the blue represents paternal, the red represents maternal, but they're the same length, centromeres at the same location, and at the same exact gene loci, geneticists found, there were 2 forms of the gene for flower color, which they eventually came to refer to as the dominant P allele, using an uppercase P to denote the dominant phenotype that Mendel had discovered, purple. For those F1 generation plants that were heterozygous dominant, they found that on the other homologous chromosome at the same gene loci, there was an alternative form to the gene that they termed the lowercase p allele. This lowercase p allele meant it was a recessive allele that coded for the recessive phenotype that Mendel had discovered, white flower color. So notice, the letter we choose is based on the dominant phenotype, which Mendel discovered, but now we apply that first letter to the genetic information, to the genes, but we call the genes alleles to denote, hey, I have a pair of alleles that code for the same trait, which is flower color. Except the trait for flower color comes in 2 forms, 2 phenotypes, a dominant purple or a recessive white. So now this right here is the same monohybrid cross, except now they're coming at you from the direction of the genetic makeup, which Mendel did not know about. Okay. So now what they're showing you is what geneticists found was the reason for how the trait for flower color was passed from the P generation to the F1 generation to the F2 generation. I said Mendel used two different true breeds. One of the true breeds, okay, always produced offspring that grew purple flowers if they self-fertilized. What they found out is the reason for that is that on the homologous pair of chromosomes at the same gene loci where you find the alleles for color, the true breed for purple flower, it was homozygous dominant. Therefore, when the cell division involving meiosis occurred, okay, 100% of the gametes would be carrying either one homologous chromosome carrying a dominant allele or carrying the other homolog, but it also had a dominant allele. For the other true breed, what they found out, the one that had the white flowers and when it self-fertilized always produced offspring with white flowers, well, what they found is that this one was a true breed because it was also homozygous, but it was homozygous recessive. Okay. On the same pair of homologous chromosomes in this plant that you find in this other true breed, you find only recessive alleles, not dominant alleles, on both homologous chromosomes. Therefore, in this plant, whenever gametes were being produced through a type of cell division involving meiosis, the gamete would either inherit a chromosome with this recessive allele, or a gamete would contain the other homolog, but it also was carrying a recessive allele. So in other words, 100% of the gametes produced were only carrying the recessive alleles. Therefore, when the gametes fertilized, when fertilization occurred between the two gametes, the F1 plants, as I said, these are hybrids. Okay, they're made up of a part of both parents, come together as these offspring. The F1 generation plants, which are our hybrids, geneticists found they were all heterozygous dominant. So they found at the level of the gene, they were able to support Mendel's findings that purple flower is the dominant phenotype. White flower color is a recessive phenotype. They were able to link that to the genotype. What they found is in these F1 plants is where they were able to officially establish that one of the two forms of the gene for flower color functioned as a dominant allele. The other gene for flower color, it functioned as a recessive allele. Therefore, when these F1 plants, just by inheriting one dominant allele, their phenotype looked just like one of the two parents, purple. However, the genotype of these F1 plants did not match either parent. They had a different genotype. They were heterozygous dominant, and they were carrying hidden information for white flower color in the recessive allele. So it's very possible for you to be carrying recessive alleles for hidden genetic information, okay, and you don't even know it. And all of a sudden, if you have children, it might resurface, it might not. It depends on the other parent. So what he did then is he allowed these F1 plants to self-fertilize. So now they're showing you each F1 plant, okay, what you would call it, in the carpel would produce eggs, and these two alleles are found on a homologous pair of chromosomes. And in the carpel, when the eggs are being produced, an egg is going to be carrying one chromosome or the other. Therefore, it's going to be carrying a chromosome with the dominant allele or the homolog with the recessive allele. Same thing in the stamen of that one F1 plant where sperm are being produced. Okay, the homologous pair of chromosomes during cell division and meiosis are going to be segregated. Therefore, some of the sperm are going to be carrying a homolog with the dominant P allele. Some are going to be carrying the homolog with the dominant recessive allele. So Reginald Punnett comes along around the 1900s, looking at Mendel's work, very fascinated with Mendel's work, and somewhere down the line, he says, you know what? I'm going to create what we call a Punnett square, okay? And I'm going to use this Punnett square to explain Mendel's findings in a much, much simpler way. But it backed up Mendel's findings. So what Reginald Punnett says is I'm going to use a Punnett square. Now, the way I do the Punnett square is I tend to extend the lines a little further. Okay, these lines, okay. Oh, there you go. I finally drew an ugly one. I was going to say, all right, they're way better than this morning, but no, they're not. I usually extend the lines a little more. If you notice over here, they're showing you sperm, and over here, they're showing you eggs. This is where you put, okay, the allelic information or the genotypic information for the gametes, sperm and the egg. Okay. Notice they're only giving you one letter, but that one letter is on a chromosome, a homologous chromosome that has been segregated from its homologous pair. In the enclosed portion of the Punnett squares, in the enclosed squares, is where we put the information for the offspring. It's where we put the information for the offspring. In this case, the F2 offspring. Notice we're showing you pairs of alleles to indicate the offspring are diploids, and they're created through the production of haploid gametes produced by diploid F1 parents. So what he says is, well, look, these F1 plants, they're all self-fertilizing. They're all asexually reproducing. Okay, so the bottom line is 50% of the sperm produced in the stamen are going to contain the dominant allele, 50% of the sperm are going to contain the recessive allele. Likewise in the carpel, 50% of the eggs are going to contain the dominant allele, 50% are going to contain the recessive allele. And Reginald Punnett says, using my Punnett square, okay, I can demonstrate there's a probability or a chance that this sperm carrying a homologous chromosome with a dominant P allele might fertilize this egg that happens to have a chromosome carrying the same dominant allele. Therefore, if this sperm by chance fertilizes this egg, I end up with an offspring that is homozygous dominant. It has a homologous pair of chromosomes, both have the dominant P allele. The phenotype I see is behind this here, is purple flower. However, Reginald Punnett's Punnett square is about probability or chance. So there's also a chance this same sperms carrying the dominant allele could fertilize the other 50%, one of the other 50% of the eggs carrying the recessive allele. If that happens, now I end up with an offspring having the genotype of heterozygous dominant. However, the phenotype is still purple, just like the homozygous dominant. But the genotypes are not the same. Again, Reginald Punnett's Punnett square is about. So it's possible one of the sperm making up the other 50% of the, okay, is carrying the recessive allele. It's the one that fertilizes the egg with the dominant allele. Now there's a probability or a chance that I end up with an offspring that is heterozygous dominant for the genotype, but for the phenotype, it's purple. So at this point, okay, there's a 1 in 4 chance that an offspring could be produced whose genotype is homozygous dominant, phenotype is purple. There's a 2 in 4 chance that there could, or a 50% chance that the offspring could be heterozygous dominant. However, the phenotypes are still purple, just like the homozygous dominant. But then Reginald Punnett says, but there's a chance that this sperm, or a probability that it could fertilize this egg. Therefore, we end up with an offspring that's homozygous, like this one up here, but it's not the same because it's homozygous recessive. So he was able to demonstrate with his Punnett square that there's a 1 in 4 chance, or a 25% chance, of an F2 offspring having a phenotype for white flower color, which explained Mendel's initial studies. It matched, it fell right into place. But instead of having to show you tens of thousands of plants, we can simply just show you a Punnett square resulting in 4 plants and the different combination of phenotypes and genotypes. So over here, for a monohybrid cross, it doesn't matter which trait we're looking at. The F2 generation always has a phenotypic ratio of 3 to 1. 3 dominant to 1 recessive. In this case, if we're looking at flower color, that one trait, it would be 3 purple to 1 white. However, notice something. The genotypic ratio does not match up with the phenotypic ratio. The genotypic ratio is described as 1 to 2 to 1. Of those 3 purple producing F2 offspring, only one of them out of the 3 is homozygous dominant. On average, 2 out of those 3 are heterozygous dominant. And then for that 1 on the phenotypic ratio, that 1 recessive, in this case for white flower color, they should always be homozygous recessive. It's the only way we'll see the recessive phenotype is if the organism is homozygous recessive. It has inherited a recessive allele from both parents. Now this one right here, everybody, in the middle, the heterozygous dominant, okay, these offspring in any organism are often called carriers. You won't see it in the phenotype as they're showing you here. However, they're carriers because they are carrying recessive information, genetic information, for another phenotype. You just can't see it. So you'll hear them sometimes referred to as carriers. So then Mendel, from his studies, okay, he was the one credited with determining there are dominant phenotypes and there are recessive phenotypes. Later geneticists came along and said there are dominant phenotypes and recessive phenotypes because there's genetic information in the form of genes, but sometimes the gene exists in more than one form. Therefore, there's genetic information that's dominant. Therefore, we refer to them as dominant alleles. And then there's genetic information that's referred to as recessive. Therefore, we refer to the genes as recessive alleles. And these dominant and recessive alleles are tied into the dominant and recessive phenotypes. And you will find that the reason that it is possible, okay, for an offspring to be heterozygous dominant, because one homologous chromosome is coming from dad, the other one's coming from mom. At the same gene loci, they will have genetic information for the same one trait, but that one trait may have more than one phenotype, such as genetic information for purple flower color versus white flower color. He formulated a law from his monohybrid crosses called the law of segregation. Okay, this law was derived from his monohybrid crosses, and it states gametes carry only one allele for each inherited trait because the allelic pairs, in other words, the pairs of chromosomes, or more specifically pairs of sister chromatids, are segregated from each other during meiosis. That's why I put this here. Do it this way. I gave you a pair of homologous chromosomes from pea plants. It's showing you at the same gene loci, I have alleles that code for the same trait flower color, except one's dominant and the other's recessive. Now imagine these pair of homologous chromosomes are represented here. When we talk about a type of cell division involving meiosis, the parent diploid cell will go through S phase of interphase, and the homologous pairs will be copied into sister chromatids. Then that parent diploid cell will enter meiosis 1. The purpose of meiosis 1 is to take a diploid nucleus and transform it into 2 haploid nuclei. Well, what we say, though, is the purpose of meiosis 1 is to segregate every homologous pair of sister chromatids, or if we want to cheat, chromosomes, to where one of the homologs gets packaged into one nucleus and the other homolog gets separated into a different nucleus. Well, imagine if they're carrying these alleles. We've just segregated the pair of alleles. Now the dominant allele is over here in a separate nucleus, and the recessive allele is in a separate nucleus over here. This is what this law is trying to say. Okay? So just take a look at those there, and like I said, watch the videos too, please. Okay, let me jump ahead here. All right. Well, Mendel, being the curious fellow that he is, he wasn't satisfied. He says, okay, I figured out the pattern of inheritance for one trait. Okay? And I figured out it doesn't matter if it's flower color, or seed shape. The pattern's always the same. If I start off with 2 true breed pea plants, one produces purple flowers, one produces white flowers, the F1 generation offspring are all going to have purple flowers. But if I take 2 true breeds, one for yellow seeds and one for green seeds, all the F1 generation plants are going to have yellow seeds because that's the dominant phenotype. But if I pull and plug and I plug in seed shape instead of seed color, and I take a true breed for round seeds and cross-fertilize it with a true breed for wrinkle seeds, all the F1 plants are going to produce round seeds. So the pattern is the same, everybody. Just pull and plug. Go back to the slide I gave you for the information on the 3 traits. Okay? And where you see dominant phenotype information for flower color, replace it with dominant phenotype information for seed color or seed shape, and then do the same thing for the rest. So Mendel noticed, okay, the pattern for each individual trait is the same. And the F2 generation always has a phenotypic ratio of 3:1. 3 dominant phenotypes to 1 recessive phenotype for every 4 plants he counted out of of thousands. So then that led to his next question. He wanted to know, okay, if different traits are linked together. In other words, if you inherit a dominant phenotype for one trait, such as purple flower color, does that mean the other traits are also going to be dominant? Does that mean then that seed color will absolutely be yellow and seed shape will absolutely be round? Or if an offspring inherits the white flower color, does that mean it's automatically linked to the other recessive traits, such as seed color? Recessive is green. Such as seed shape, recessive is wrinkled. Are they linked? Or are these traits passed on independent of each other? In other words, are they not linked? Is it possible for an offspring to inherit a purple flower color, a yellow seed color, but it has a recessive shape wrinkled, or any combination of those? This is the purpose of his dihybrid crosses, okay? To establish if different traits, the dominant phenotypes for different traits and the recessive phenotypes for different traits are linked or not linked. Therefore, he performed his dihybrid crosses. Listen carefully. The cross means the same thing as in a monohybrid cross. We begin with the P generation, where Mendel produced two different true breeds, okay? But he cross-fertilizes the true breeds to produce an F1 generation. The hybrid is in reference still to the F1 generation. These F1 generation offspring are hybrids coming from two separate parents, okay, who sexually reproduced or through cross-fertilization. But the die, the way I define the die, focuses on two different traits. That's how I define the die for dihybrid cross. Die means focuses on two different traits. Examples: seed color, seed shape. Now, this is the only example of a dihybrid cross I want you to know, okay? Only these two traits, seed color, seed shape. For monohybrid, I could ask you about flower color, I could ask you about seed color, I could ask you about seed shape, but understand it's the same pattern, just pull and plug. For a dihybrid cross, I won't do that. I will only use this example, and there's a video link for this. Okay. So Mendel performed his dihybrid crosses because he wanted to know if the different traits were linked. You know, our dominant phenotypes for different traits always passed on together to an offspring. Our recessive phenotypes for different traits always passed on to offspring. Or can an offspring inherit a mix of dominant and recessive phenotypes for different traits? So the two traits I want you to focus on for this going into the test: seed color and seed shape. Now, Mendel proposed two hypotheses. Okay, if dominant phenotypes for different traits and recessive phenotypes for different traits are linked together, then his first hypothesis he coined dependent assortment. In other words, if you inherit this dominant phenotype for this trait, then every other trait that we look at is going to also be a dominant phenotype because they're dependently assorted. They're always passed on together. However, he had a second hypothesis, and I don't know if I misspelled it in your notes. It shouldn't be SIS, it should be SES if we're talking plural. But he proposed the second hypothesis. He called this one independent assortment. What he meant here is if you study two different traits, such as seed color and seed shape, is it possible to see the dominant phenotype for, say, seed color passed on, okay, and then the recessive phenotype for seed shape being passed on? In other words, the two traits aren't linked together. So it is possible to inherit both dominant and recessive phenotypes for different traits. So they're showing you here two columns, okay? But the truth of the matter is he starts off his dihybrid cross with two true breeds. One of the true breeds, okay, if it self-fertilizes, always produces round yellow seeds. Both of those are dominant phenotypes for two separate traits. And he cross-fertilized it with the true breed that if it self-fertilizes, always produces green wrinkled seeds. Okay, so you can see here they're showing you two allelic pairs for both of them to indicate two different traits. These are the same here. They're just showing you in two columns under each hypothesis. When Mendel took his P generation and cross-fertilized him, okay, his results ended up being what he anticipated. The F1 generation were hybrids that always produced round seeds that were yellow. Okay. Then what happened is that he knew in order to prove and disprove these two hypotheses, he wouldn't be able to do that until he produced the F2 generation. Because just like he found out with the monohybrid cross that some traits can be hidden and skip a generation, he knew that the answer to his question about two different traits are dominant and recessive phenotypes always linked together and passed on together. He knew he would get the answer to that with his F2 generation for the pea plants. So just like he did for monohybrid crosses, it's the same pattern. From P generation to F1 generation, we take two separate parents, two true breeds, cross-fertilize them and produce our hybrid F1 generation offsprings. But in this case, to see if the traits, two different traits are linked, he did it again, just like with monohybrid crosses. He took each individual F1 generation offspring, allowed them to self-fertilize. Now, he had predicted that if this hypothesis dependent assortment was true, he predicted his F2 generation would result in a phenotypic ratio of 3 to 1. On average, for every 3 pea plants in the F2 generation he analyzed, okay, 3 would be producing round yellow seeds. And on average, for every 4 pea plants from the F2 generation he analyzed, 1 out of 4 would have wrinkled green seeds, both recessive phenotypes. However, he knew if the second hypothesis was true, his F2 generation, okay, there would be a greater amount of genetic variation. Therefore, there would be a greater amount of phenotypic variation, and that's exactly what he got. So listen carefully. For dihybrid crosses, it wouldn't matter which 2 traits we would focus on out of the 7 total that Mendel studied. Okay, for the F2 generation dihybrid crosses, he always had a phenotypic ratio of 9 to 3 to 3 to 1. So again, for Mendel's dihybrid crosses, it doesn't matter which 2 traits he was focused on. For Mendel's dihybrid crosses, his F1 generation always had a phenotypic ratio of 9 to 3 to 3 to 1. Now, what does that mean? Well, he produced tens of thousands of plants, and on average, for every 16 plants he analyzed, 9 out of 16 produced both dominant traits. Yellow colored seeds that were round, those are both dominant traits for 2 different traits, seed color, seed shape. Jumping down here to the bottom, on average for every 16 plants he analyzed, 1 would produce seeds that are green and wrinkled. Both of these are recessive traits. Green is recessive for seed color, wrinkled is recessive for seed shape. It was these 2, the 3 and 3. It was these 2 groups that proved his second hypothesis true, independent assortment. Okay? Because these 2 groups had a combination of a dominant phenotype combined with the recessive. So the first 3, okay, it represents green and round. On average, for every 16 plants that were produced, on average, 3 of them produced green seeds, which is recessive color, round seeds, which is dominant shape. So there we have a recessive color with a dominant shape. We have a recessive phenotype with a dominant phenotype. And then the other ones, on average, there would be 3 out of 16 offspring that would have the dominant color for yellow, but the recessive shape for wrinkled. So it was these 2 groups that proved independent assortment. It is possible to inherit dominant phenotype for one trait, but a recessive phenotype for another trait. Now, notice in here though, they're giving you the genotype combinations. So even though we have 4 different phenotypes at a ratio of 9 to 3 to 3 to 1, if you look in here, there's a greater variation in the genotypes. Okay, it'll be up to you to go through that, okay? On the test, I will test you on dihybrid and monohybrid crosses, but for dihybrid cross, this is the only example I will use. Okay, make sure that you go over the letters here, which represent the alleles and how it relates to the color and shape. And then notice up here on the Punnett square, they give you the possible combinations of alleles in the gametes or haploid sperm. And down here, they give you the possible combinations of alleles in our haploid gametes, or more specifically, eggs. They put it together in a Punnett square and show you the probability that this sperm could fertilize any one of these 4 eggs. This sperm could fertilize any one of these 4, and same for the last 2. Okay? I would not ask you to, I may ask you to fill out a monohybrid cross, a Punnett square for a monohybrid cross. I will not ask you to fill out a Punnett square for a dihybrid cross. I would give you some information, and then it would be up to you to answer the questions. So from his dihybrid cross, he came up with the law of independent assortment. It came from his second hypothesis, and I'm going to let you read that. But what they're showing you here, if you notice, has to do with the type of cell division we talked about in chapter 13. Cell cycle involving meiosis, okay? So I'll let you look at this, and I'm just showing you where the monohybrid cross applies for law of segregation, and where the dihybrid cross, he derived his second law of independent assortment. So I'll let you look at that, and notice it says 2 different pairs of alleles. They're showing you up here 2 different pairs of alleles, R's and Y's. It's just a matter of whether they're lowercase or uppercase. That's why I'm pointing up here also and down here, and I'm not doing it here. Okay? So remember, dihybrid focuses on 2 traits, so we're looking at 2 different pairs of alleles. Monohybrid focuses on traits, so we're looking at 1 pair of alleles. So again, Reginald Punnett's Punnett square is about probability. Okay, and I'll let you look at that. I'm going to jump ahead because we're running short on time. So there were those that are interested in animals, especially animals that, like I said, we don't say true breeds, we tend to say pure breeds. And these breeders, a lot of them, or the best ones, are well versed in genetics. Okay, I was watching a documentary on that. I was like, oh my God, this guy's way the heck ahead of me. He's applying the genetics he learned and going beyond it. He was breeding animals. But what happened is, as these breeders became familiar with genetics, they realized they could run what we call test cross to determine if certain traits were due to the animal being a pure breed or the animal's not a pure breed. Case in point, for Labradors. Labradors, it depends on which species, or if you want to call it which group of Labradors you're talking about. But there are Labradors that sometimes they're called black labs. Okay, they have a black coat color, and that black coat color happens to be the dominant phenotype. However, you don't hear that there's a recessive phenotype in relation to these black labs. It looks brown, but they call it a chocolate coat color. The chocolate coat color is recessive. So breeders know that when they're looking at this type of coat color for labs, if they see a Labrador that actually has a chocolate coat color, they don't even have to take it in for a genetics test. They know it's homozygous recessive based on previous genetic studies. However, these breeders also know that if they're looking at a black lab, it has the dominant coat color, black coat color. They know, hey, I don't know if it's a pure breed or not. If it's a pure breed, it'll be homozygous dominant. But if it's not a pure breed, it's going to be heterozygous dominant. So what they would do is a test cross. We'll take a lab with the chocolate coat color because we know it's pure. It's homozygous recessive. It's pure breed. Okay? And we'll have it mate with the lab that has a black coat color. And if this lab's black coat color is due to it being a pure breed, in other words, it's homozygous dominant, then all their offspring will have a black coat color. However, if this black lab is not a pure breed, it's got a black coat color, but it's heterozygous dominant, it's not a pure breed, then some of the offspring should have that chocolate coat color inherited partly from this other parent. And that means this one is not a pure breed anymore. So this is one way of doing it. Now, obviously, genetics testing made large advances, but it also depends on money you have. But this is one way they can determine for a particular trait, one trait, whether or not the animal in question is a pure breed or not. Notice, if they're pure breed, they'll always be homozygous, everybody. Or, I'm sorry, homozygous here. Now, geneticists also came out with what we call a pedigree. I define a pedigree as a genetic family tree. So again, I define a pedigree as a genetic family tree that allows you to track a trait from one generation to the next. Okay, so a pedigree is a genetic family tree. You can see this is what they're showing you here. It looks like a lot like a family tree, but it's a genetic family tree that allows you to track a single trait from one generation to the next. They will use shapes to represent males and females. So squares represent males circles represent females. And then since the purpose of a pedigree is to actually track a trait from one generation to another, okay, whichever person has that trait of interest, we shade it in. We color in the shape to let you know, hey, look, okay, this is my pedigree, and these individuals are the ones that have the trait that I'm interested in here. Okay. Now, the way it works is that they'll start with the oldest generation at the top, okay, and they don't have to be married. What they'll do is they'll draw a line connecting the male to the female, and then they'll draw a line coming down saying that they had children, they had offspring. Whether or not they're married, we don't know, but they had children. And then if they have more than one child, what they do is they list them from oldest on the left to youngest on the right, and you can see how this one line branches out connecting them all. And then if any of those children, okay, end up having their own children, they'll draw a line connecting them, okay, to whoever it is that they had an offspring with. If it's just one, they'll put a line here. If it's more than one, then they'll split the line, going oldest to youngest. And then if that one of those two children has children, then they'll connect it. And then they'll look at each generation saying, who has the trait I'm interested in? And they'll shade it in. These are some of the traits you can track. Now, I put a video for pedigrees next to pedigree, and I believe it's earlobes that they track. Okay, but these are what they're doing it for. More often than not, when a pedigree is put together, it's not for a good reason. It's to track a trait that's led to a disease, a disorder, an illness. Deafness. There are many different causes for deafness. Okay, one cause of deafness is due to a recessive allele. And what they've done here is they've taken Jonathan, who is deaf, and his wife Elizabeth. They ended up having a lot of kids, 7 kids by today's standards, not by my parents' standards, but by my standards. They had 7 children. 2 of them are deaf. The other 5 had normal hearing. So what ends up happening is sometimes what a doctor will say is, well, let's put together a pedigree, a genetic family tree tracking the trait, in this case for deafness. And then what they'll say is, hey, let's start with, say, the grandparents back here. Okay. And here they're showing you the grandparents. They didn't fill in the circles or the squares, meaning that they're not deaf. Below it, what they try to do, in addition to giving the names, they'll try to give you the allelic pairs associated with the trait that they're tracking. Okay. Now, sometimes, even if the person in this case has normal hearing, if they know that the trait they're tracking is due to a recessive allele, they may have to put a blank. In other words, I can't give you the allelic pair that these 2 individuals have because I can't tell if one or both of them are carriers. Okay. However, down here, they're saying that the daughter from these 2 is a carrier. So the reason why they have a blank here is because, like I said, they don't know if one or both of them themselves are carriers. So they'll try to fill in the allelic information and put together to see if on paper, if the cause of deafness might be due, okay, to traits being inherited through your genetic information. And if they can establish a relationship on paper, then the doctor will go further and say, okay, now let's see if we can actually detect the actual allele to validate what we see here in the pedigree. And they'll do this for other types of genetic disorders such as albinoism or dwarfism. There's many different causes of dwarfism, but one type in particular called achondroplasia. It's a dominant genetic disorder determined by a dominant allele, whereas albinoism, it's a recessive phenotype associated with the recessive allele. So I'll let you take a look at these 2 here as I move on, because I won't go real in depth into testing you on anything. Okay, extensions of Mendelian genetics. So as I said, Gregor Mendel is considered the father of genetics. He established a foundation for the field of genetics. Now, that doesn't mean that geneticists, other scientists who have gone on to study other species haven't come up with results that differ from Mendel's, but they have something to compare it to. Okay. Examples, I'm sorry, I'm starting to choke here, so I'm about to stop. So for example, a dominant allele. Is there some times where a dominant allele isn't completely dominant? And is it possible that there could be not 2 possible alleles, but say 3 possible alleles for the same phenotype? The answer for both is yes. What geneticists discovered later studying other species is sometimes a dominant allele is completely dominant. it definitely determines the phenotype. Sometimes the dominant allele, okay, if only one dominant allele is inherited, results in an intermediate phenotype. And then sometimes there's more than one dominant allele along with the recessive allele. In other words, there's at least 3 different alleles for the same phenotype. And I'm going to go a little bit over time, so bear with me here. So what Mendel's found in pea plants in regards to all 7 traits was what we call complete dominance. That's where any offspring that are heterozygous dominant, okay, only the dominant allele completely determines the phenotype. Okay, so the dominant allele codes for the dominant phenotype. But in other plant species, such as snapdragons, what they found is it's not always true. There are species of snapdragons where red flower color is dominant and white flower color is recessive. However, when you perform a monohybrid cross, the F1 generation give you an intermediate phenotype, which they refer to as incomplete dominance. Well, what it is in some species of snapdragons, there's an allele that codes for a red pigment. And when an offspring inherits 2 of these alleles, you get a very high concentration of this red pigment, therefore the flower looks red. However, there's another allele, a variant, that codes for white flower color. In other words, the alleles contain genetic information that doesn't code for any color, so the default color is white. So when an offspring, such as an F1 offspring, is produced through cross-fertilization between these 2 parents, the offspring inherits 1 dominant allele for red and 1 allele for white. But what happens in this case is since it only inherited 1 red allele, it only gets half as much red pigment being produced compared to one of the parents. Therefore, we get what we call a pink color, but you could also think of it as a lighter shade of red, incomplete dominance. Just this last one, please. And then ABO blood typing. What they found for ABO blood typing is, first of all, your ABO blood type is genetically determined by what you inherit, by the alleles you inherit from your parents. The catch is there are a total of 3 different alleles, 3 different genes that are responsible for your ABO blood type. You will only inherit 2 out of these 3. One of them is recessive, and that's the one that's tied into blood type O. But the other 2 alleles are both dominant alleles, so they denote them by uppercase I with a superscript A or B because the phenotype they code for varies. Okay, now we say antigens, but here it says carbohydrates, but often you'll say antigens. There's a dominant allele that codes for antigen A, and there's a dominant allele that codes for antigen B. So somebody who is type AB, okay, their phenotype is demonstrating codominance. Anyone that has type AB blood, what has happened is you've inherited 2 dominant alleles, but from one parent, the dominant allele is for A, and the other dominant allele is for B. Therefore, on your red blood cells, you end up with both phenotypes being equally expressed, which we refer to as antigen A and antigen B. So sometimes there's more than one type of dominant allele. They both equally impact your phenotype. Okay, yes, I'm stopping right there. I'm barely hanging on here. Sorry for going a little overtime. Let me stop the recording.